Chapter 20, Vascular Physiology, Screencast 1. This series of screencasts will be focusing on the physiology associated with blood vessels. This will include topics such as what's happening at the level of the capillary, blood pressure, local flow, the regulation of blood pressure. So those are the things that we're actually going to be covering in this series. Now, the first slide that we're going to take a look at, this is a really important image. And what we have here is the relationship between total cross-sectional area and the velocity of blood as it's moving in vessels. Now, unsurprisingly, if we take a look here, okay, in the arteries and the larger arteries in particular, we see the greatest velocity. As we start to get further away from the heart, we see a decrease in velocity. Now, something else that we're looking at as we start to get to other sets of vessels, we start to see an increase in the cross-sectional area of the vessels. And take note, the vessels that have the greatest amount of cross-sectional area are capillaries. Now, remember, the role of capillaries is actually exchange, and that goes both ways, the delivery of nutrients and the picking up of metabolic waste. And when we look at what's happening, the velocity in the capillaries is actually quite slow. Now, this is also really important because what we're going to look at here, because the velocity of blood moving through the capillaries is slow, this is going to maximize the amount of time that is available for exchange. So that's really important, especially when we're looking at the role of capillaries. And then basically, as we leave the capillaries and make our way back to the venous side, we start to see a bit of an increase in velocity, but notice not nearly as great as what we saw on the arterial side. Another thing to keep in mind here is when we are looking at flow through capillaries and veins, it is continuous, unlike what we see in the arteries, which is pulsatile. So that's a really important difference between the two. Now, just a quick review of some transport mechanisms. Primarily, we're looking at diffusion and vesicular transport. Now, remember, when we're talking about diffusion, we're basically looking at substances that are going to be either leaving the blood or entering a blood and going down their concentration gradient. So remember, this is a passive process, and things will be moving from an area of high concentration to low concentration. Examples of things that are going to move into the interstitial fluid will be oxygen, hormones, nutrients, while things that are going from tissue to blood are going to be carbon dioxide and other waste tissues. Now, depending upon the size of the particle, we're going to see different pathways. Solutes that are small, they can actually move through the endothelial cells or the intercellular clefts. This goes back to what we said previously when we talked about tight junctions. Tight is not perfect, and there are gaps that things can actually move through. When we start looking at larger solutes, so example, proteins, they're going to only pass through structures or vessels that have fenestrations or gaps in the sinusoids. Now, when we look at vesicular transport, right now, we're basically looking at pinocytosis and exocytosis, and this is what the endothelial cells use. Now, when we talk about pinocytosis, we're actually looking at a fluid-filled vesicle that formed at the plasma membrane via an invagination. Then that transport vesicle actually makes its way across the cell. Now, when we look how things are going to be excreted, that is going to be exocytosis, and that's another example of vesicular transport for the most part. So keep in mind, we're looking at things moving in both directions, and that's going to be important. And the substances that are most usually transported in this particular manner are going to be some hormones and fatty acids. Now, The next topic that we're going to address is going to be really important, and I'm going to do a separate screencast describing this topic as was done in class. It's not going to be included in this one. There'll be a separate screencast addressing this concept. So now we're looking at bulk flow. So this is going to be fluids moving down a pressure gradient. So we have a hydrostatic force here. Now, keep in mind, we're really looking at large amounts of fluid, but it's just not fluid. We're also going to see substances that are actually dissolved in that fluid move along also. Now, what's going to determine the direction of the movement is going to be the net pressure of opposing forces. I want us to think of this as a tug-of-war. Now, the two forces that we are going to be looking at in particular are going to be hydrostatic pressure and colloid or oncotic pressure. I actually prefer the term oncotic because it is more specific to the force exerted by protein, and that's really what we're addressing here as protein being a non-permeable solute. Now, when we talk about filtration, that's going to be fluid moving out of the blood. So what we're going to see is basically fluid and small solutes will easily move through the openings within the capillaries. Generally, this is happening on the arterial end of the capillary, whereas reabsorption is going to be the opposite of that. We're actually looking at fluid moving back into the blood. And once again, we're really talking about capillaries at this point in time. Now, reabsorption is actually going to take place on the venous end of the capillary. Now, as I indicated previously, we're going to have two different types of forces. The first set of forces we want to address are going to be hydrostatic forces. And when we talk about hydrostatic forces, what we're really talking about is the force that a fluid will exert against the wall. Now, keep in mind, we're going to be looking at blood hydrostatic pressure. I quite often will refer to this as capillary hydrostatic pressure. So this is the actual force exerted by blood on the wall of the capillaries. This is going to be a force that actually favors filtration. So fluid and substances moving out of the capillary and into the interstitial fluid. When we look at interstitial fluid hydrostatic pressure, we are basically going to think of this as zero. And the reason why, basically, this would be the force attributed to fluid in the interstitial or outside the vessel. Now, think about what's going on here. What we're actually going to see is in the interstitial space, there's a lot of space for that fluid or the force of that fluid to basically disperse. Also, there's going to be some other mechanisms that are constantly happening that are going to play an important role in actually picking up fluid out of the interstitial space. As I said, when we look at interstitial fluid hydrostatic pressure, we're basically going to assume that's zero. Now, another important thing, though, that I want to go back to and talk about blood hydrostatic pressure, that is going to decline as we move from the arterial end of the capillary to the venous end. That's really important. So keep in mind, both of these forces are going to exist, but when we have a healthy individual, the only one that we're going to put an emphasis on is capillary hydrostatic pressure. And remember, this will decrease as it moves from the arterial end of the capillary to the venous end. The next set of forces that we have are colloid. I actually like to prefer or call this oncotic. This is a term that's more specific when we're talking about the force exerted by protein or non-permeating solute. Now, what we're going to find here, that this is going to be a force that actually favors reabsorption. So we're going to think of it as a suckage force. Now, keep in mind, really what we're talking about, the movement of water, is osmosis. And how I like to describe osmosis is basically water follows solute if it can. And we're going to think of protein as a non-permeating solute. Before we really talk about these forces, I want to talk about where we expect to find proteins. Now remember, proteins are large, and we are going to describe them as a non-permeating solute. So what's important here is to keep in mind these proteins are found in two places. They're going to be within the plasma, so in our vascular system, which is closed, and also cells. So what we're going to find here, basically, proteins are in what I'll refer to as a container. It could be the cell, or it could be a completely enclosed system. And once again, they're considered to be non-permeating solute, so they're going to draw water towards them, and that is really what we're talking about, the force that's going to drive reabsorption. Now, we're basically going to assume that this is constant. And the other thing that we have to remember here is it's due to the proteins that we have, and these proteins are actually produced by the liver. So once again, this is going to be the force that primarily opposes capillary hydrostatic pressure. So keep in mind, we're really looking at a tug of war. Now, the other pressure that we have to address is interstitial fluid colloid osmotic pressure. This is going to be a force that draws fluid into the interstitial fluid. But remember, we don't expect to find proteins in the interstitial fluid, so we're going to think of this as a non-player in a healthy individual. So let's take a step back and think about the two forces that we really have to worry about. We have to worry about capillary hydrostatic pressure, and that will decrease as we move from the arterial end of the capillary to the venous end. And then we have capillary oncotic pressure, and we are going to assume that this is constant. Now, what we have to look at here that's determining the direction is going to be the net filtration pressure. I'm not going to spend a lot of time going over these calculations, and you are not responsible of calculating this. But really what we're looking at, net filtration pressure, is the difference between net hydrostatic pressure and the net colloid osmotic pressure. So let's keep in mind, we're really looking at that tug of war that's happening between capillary hydrostatic pressure and capillary oncotic pressure. Now, if we look at net hydrostatic pressure, that's the difference between blood and interstitial hydrostatic pressures, whereas the colloid or oncotic pressure is the difference between capillary oncotic and interstitial fluid osmotic pressure. So keep in mind that this net filtration pressure will change along the length of the capillary, and here's where the tug of war comes in. And what we're going to notice that at the arterial end, when capillary hydrostatic pressure is greater than capillary oncotic pressure, this is going to be filtration, and filtration generally happens primarily at the arterial end of the capillary. Now remember, this capillary hydrostatic pressure declines as we move across the capillary bed. So we will get to the point when capillary hydrostatic pressure is less than capillary oncotic pressure. So now this will favor reabsorption, and it's the force exerted by proteins pulling fluid back into the capillary. Now, I find this diagram to be a little confusing because we're really looking at numbers, and I don't want us to focus on the numbers. As I said, I'm going to do another recording that will focus on how this was covered during lecture, and I think that is a little bit easier to understand. But really what we're looking at here is basically as far as that hydrostatic pressure, and we basically see that as long as capillary hydrostatic pressure is greater than capillary oncotic pressure, we're actually going to be seeing filtration or fluid moving out of the capillary. When capillary hydrostatic pressure is less than capillary oncotic pressure, we're now in a situation that favors reabsorption and fluid will move back into the capillary. That's really what it is. It's just that tug of war between capillary hydrostatic pressure and capillary oncotic pressure. Now, one thing that we have to keep in mind that is especially important, there is some excess fluid that's not reabsorbed completely into the capillary at the venous end. This accounts for about 15% of fluid not reabsorbed. Now, this is something that's really important because we really can't afford to lose 15% of blood volume. So there has to be a mechanism that is going to result in saving this fluid, or somehow it's working its way back into the vascular system or back to blood. And this is going to be where the lymphatic system comes into play. There's two major jobs associated with the lymphatic system. One is fluid balance, and that's what we're addressing here now. So this fluid is picked up by lymphatic vessels out of the interstitial fluid, and it's going to make its way back into the subclavian veins. And that's how this fluid returns or gets back into circulation.